Thank you very much, and thank you for inviting us to talk about this interesting and really important topic in our research. So we have a very eminent set of panelists here today who are some of the leading regenerative medicine and stem cell model systems experts. So we have Andrea Brand from Cambridge University, Elly Tanaka coming to us from Austria, and Allison Bardin coming to us from the Curie. So you see model systems pervade the whole planet, and really what I want to try and just do in my brief introduction is give you some conceptual framework of why we use these systems so the science that these eminent scientists are producing can shine and inform on why we use them. So model systems are really important in terms of stem cell biology because most of the concepts that we study that we endeavour to envisage employing in human systems have really been developed over the last several decades using model systems. And I think we tend to approach science naturally from an ego-driven perspective, as this slide puts forward, that we seem to consider ourselves the pinnacle of creation often, and then the systems and techniques or processes that our body uses are really fundamentally superior or more important. But of course, nature does not work that way. And the way that stem cell actions are best understood are in the context of basic principles that can best be derived from model systems. So there are a number of reasons why we use animal systems in stem cell research. Chief amongst those, of course, is we can't really experiment on ourselves, and we can't really see stem cell action in living tissues in our own body. But there's a fundamentally more interesting and important aspect of that, is that they tell us about general principles. It's not just about knowing how stem cells act in particular contexts in our body, but how do stem cells act generally throughout the phylogeny to generate the tissues and repair systems that we know about. And also, of course, although some of our organ systems are regenerative, many of them are not. And many of our speakers today will talk about pro-regenerative capacities that animals have that we do not. So it's much better to have a roadmap of trying to achieve regeneration than try to derive it from first principles. So that is an important concept for model organism systems. So also the other thing that are really critical in all the systems that we're going to talk about today is genetics. The modern era of genetics are the real framework for evolution and biology. This is the golden age of biology. It was chemistry and then it was physics, and now it's biology. Genetics has underpinned most of our understanding of the way, the molecular framework, how do things actually function to derive tissues and organs and stem cells. So I've just illustrated a couple of things about what makes a good model system and why we use the ones we use. Really, the Rolls-Royce of model systems has been Drosophila for many years, and as you can see, the candidates for model systems have to have unique attributes. So good genetic systems have to be bred in large numbers, have short generations time, and so you can do genetics in a period of time that is tractable. So other organisms need to be, that study embryology and the way that stem cells work in the embryo, their embryology has to be accessible. And finally, in the area of modern genomics, probably hear a little bit about this from Elly, the genome rules supreme. You must have access to the genomic information of model systems, and so having access to the genome is important. So this has all come together and produced an enormous interest in model systems. And as I mentioned, one of the things that's really kind of critical is that many of the systems or models that we use can regenerate tissues and organ systems that we cannot ourselves. So the question we simply ask in those systems, what capacities or what processes do these systems or animals have that we do not? And so we'll probably hear a bit about that from Elly as well. So all of this has come together, the fact that we have model systems, we have modern genetics, we have genomes, in a sort of perfect storm of activity that allows us to unlock the potential of using these animal systems to understand regeneration biology. And this is just a simple chart showing that there's been an enormous rise in the number of publications using model systems, various ones going quicker or not going quicker. But you can see that the number of publications on the left have risen dramatically in the last couple of decades, and such that this is a widely adopted approach for using model systems. My own system, the zebrafish, is a fairly humble organism, and its major feature and capacities that enable us to image stem cells is that it's optically transparent, and it combines all the genetic activities I've talked about before. So this is just to illustrate the power if you've got an optically clear embryo and you can image it, and we're going to see some gorgeous images, I know, from all the speakers today, what that lets you able to access. And these are just some what we call lineage tracing, follow the progeny of stem cells over time. And this allows us incredible depth and capacity of understanding the way that stem cell systems live in living tissues, if we can actually see them. And here is just some images again of stem cell activity in living tissue. Normally this would be something you'd see down in a culture dish, but these systems now allow you to look at interactions between stem cells and their environment and really try to understand very deep activities. So what I hope we hear from our speakers today are illustrations of the attributes that I've tried to say make a good model system, and try to convince you that these are where the really deep understandings of stem cell action are made and understood. And then, of course, we look in more complex systems or more difficult scenarios, such as our own biology, to see if those general concepts hold. Right, so without that, I believe, Andrea, you're next.
So one of the things that we're interested in my lab is whether it might be possible in the long term to be able to repair the brain after damage or disease. And obviously this is something that is of great concern as people are aging, and we hear more about various types of dementia. So it would be wonderful if we had enough understanding of how the brain is put together and how it functions to be able to repair it or regenerate it after any kind of damage. Now, as Peter was saying, stem cells are the origin of most of the different cells and tissues in the body. And in fact, the brain and the nervous system arises from neural stem cells, and they give rise to all of the different types of cells, neurons and glia, within the brain and the entire central and peripheral nervous system. Now, the interesting thing that's been discovered over the last 10 or 20 years is that these stem cells produce the neurons in the brain and nervous system, not only early in development during embryogenesis, but there are also stem cells in the adult brain. And it's thought that these stem cells are important to maintain the brain for homeostasis and repair. But what's intriguing, if you think about stem cells in the adult, is that most of these stem cells are dormant. They are not proliferating, they're not generating new cells. And this is true not just of the stem cells in the brain, but also in the stem cells of other tissues and organs, for example, in the skin, the mammary gland, in the intestine. So what we're interested in is this dormant state, and indeed, most importantly, is there some way of waking these stem cells up? Because if you could wake them up, then they can proliferate and produce new cells, which could be important for repair of organs and tissues in the adult. So the question that we've been addressing in our lab is, how are these stem cells awoken? Now, we know some things about how, what sorts of stimuli will wake up stem cells, and these are various things from, for example, nutrition, exercise, or in fact, injury or disease. And these sorts of stimuli can push these stem cells out of this dormant or quiescent state to allow them to start dividing again and produce new neurons. Now, the pictures I've been showing you have been of human beings, but as Peter mentioned, it's very difficult to study some of these processes within human beings. And in fact, if you look at the nervous system in human beings, it's very large, very complex. So 100 billion neurons and a quadrillion synapses or connections between the neurons. And in fact, it can be difficult to identify the stem cells in human tissue. So for these reasons, we'd like to study a much simpler system. And the system that we chose to study is the fruit fly Drosophila melanogaster. And so by comparison, the nervous system is much simpler. There are 100,000 neurons in the fruit fly compared to 100 billion in man. But what's perhaps key, most important, is that if you look at all the genes in human beings that are associated with various diseases, about 75% of them have counterparts in Drosophila. And so it means that we can study these genes and understand how they work in a very simple system, but what we learn in the simple system can then be applied to higher organisms. So I love this picture. This is one made by Andreas Prokop from the University of Manchester, just showing how very similar the human nervous system is to Drosophila. And what I'd like to show you here is, they both have brains. This is a Drosophila larva, and here's the spinal cord, here's the ventral nerve cord in flies. But I just want to point out they're not to scale. You're not going to meet a Drosophila larva walking down the street, so don't worry about that. But there are a lot of similarities, even though the body plans don't look that similar to one another. So what we've been able to find a few years ago working on Drosophila is that one of the key signals to wake up stem cells is in fact insulin. And insulin, this is secreted within the brain in response to nutrition. So remarkably, a rather similar system to what you know about the pancreas, but here we've got insulin within the brain waking up the stem cells. And as I said before, what we'd really like to believe is that if we can understand about the basic regulatory mechanisms that regulate stem cells in the brain, then it should be possible in vivo within the animal to wake up the stem cells and allow them to generate new neurons as a way of repairing or regenerating the brain or the nervous system after damage or disease. Now, this is another key thing to remember, though, is that while we'd like to be able to wake up the stem cells and produce new neurons, one has to be a little bit careful because you don't want too much proliferation. And in fact, if there's too many cells dividing in the brain or in other tissues, it can lead to cancers. And this is another thing that we can study in the fruit fly, because you can see here a brain tumor within the fruit fly brain. And so what we're trying to understand is how to get these cells to proliferate when we want them to, but how to prevent them from over-proliferating and giving rise to tumors. And this very simple model organisms allows us to follow both of these processes. And I'll stop there and hand over to our next speaker.
Thanks, Andrea. So I think our next speaker is Elly Tanaka, who's going to tell us about the wonders of regeneration in the axolotl and salamander system.
Okay, so I'd like to talk to you about the axolotl system, which is really one of the most spectacular examples of regeneration in a vertebrate who has limbs. So really a vertebrate that's a tetrapod that really uses limbs like we do. But thinking about limbs, we know that, that the ability to repair or regenerate limbs could really help humans. But of course, since humans don't do this, one wonders, is the quest to regenerate a human limb more like alchemy, or is it really an engineering problem? So, of course, we know that alchemists, for a long time were trying to make gold, but were never successful. And that if we think back, that the molecular principles of this endeavor really were not sound. But on the other hand, if we think about the process of human flight, we were successful. So in this case, this is a picture from a book by Otto Lilienthal, who was really obsessed with human flight. And here he studied how storks use their wings to fly. And this was very important for him to start designing wings, human-sized wings, for the attempts at flight. Is this going forward or, oops. I need someone to, yes, okay. And this is a picture of him using this information to undergo flight. Now, these attempts were, and these taking lessons from nature and these attempts were extremely important, although he himself was not the person who might be considered the person who made the first successful airplanes. But Orville and Wilbur Wright used his information along with other information from his studies to design then next advanced wings that torqued in a way that was then worked better for sustaining flight. And of course then airplanes then attach onto that motors and other artificial things to sustain this flight. So we have now airliners that are highly artificial but do allow us to fly and do still use the principles learned from birds. So we think then of the salamander as this natural inspiration that can teach us some general principles of how a limb that has very much the same structure as our limbs can regenerate. And so we're very intensely studying this model system, using all the information from our recently acquired genome, using many ways of tagging molecules and cells in the body to understand how an example of successful regeneration occurs. Oops. Okay, can you advance the slide? Yeah, thank you. So I wanted to illustrate one or two examples from our research where we've identified molecules that are present throughout vertebrates and are important in the process of regeneration. So, for example, we've identified two proteins that are present in the blood of all vertebrates that are very important for triggering the process of regeneration. This is a schema of a limb that has been amputated, and the skin cells have crawled over the end of the limb. What we found is that when platelets aggregate at the wound site, they release a protein called platelet-derived growth factor that we've shown is essential for the cells in the tissue to start migrating to the amputation site. And this migration to collect cells at the amputation site is absolutely essential for the process of regeneration. In addition, we found that there's a protein called bone morphogenetic protein that's circulating in our blood vessels in the blood. And when, with the amputation, the blood vessels are broken and these proteins are released into the surrounding area. And not only are they released, the proteases that cleave are part of the clotting process also cleave these bone morphogenetic proteins and make them highly active. And these proteins induce the cells that have accumulated at the amputation site to proliferate. Now, we've also worked on other assays of later parts of regeneration based on some very curious biology that had been discovered by a number of researchers in the field. This shows that when you take the regenerating tip of the regenerating tip portion of a limb of an axolotl and transplant it from the left side limb to the right limb, you get actually 3 limbs growing out of this chimeric structure. This gave people information that meant that the cells on the thumb side of the limb of the arm are different than cells on the pinky side of the limb, and that the animal uses this information to then direct where a new limb should form. By using information gathered from people studying the development of the limb, we were able to take an educated guess of what factors might be produced in the pinky side of the limb that are important for inducing a new limb to grow. So using information from previous researchers who showed that if you redirect a piece of nerve to the side of the limb and then provide pinky and thumb tissue here, you could grow an entire limb out of the side. So then we made this educated guess that this factor called sonic hedgehog could be sufficient to induce an entire limb to grow in a place that we've innervated by rerouting nerves. And this just shows you the sample of this limb that's grown out by the treatment of these animals with this factor called sonic hedgehog. So the lesson here is that by studying salamander regeneration, we've identified an important role for several factors, and all of these factors are present in humans. So, of course, we're currently trying to understand why human cells don't undertake these responses as fully as salamanders do. And we think that future regeneration attempts may induce something like a full limb to regenerate in humans may involve hybrid approaches. On one hand, we could stimulate endogenous cells with such factors. It's obvious we need still to identify other blocks that may be existing in humans. But then, of course, we may bioengineer and transplant pieces of competent tissue that we've engineered in vitro to help the process along. We may even be able to substitute certain aspects of the limb function and limb regeneration with purely material engineering. And of course, these things, they then may interface with modern prosthesis. So just thinking about the airplane model, we do believe that we may end up with combining approaches to engineer complex regeneration in humans. Thank you very much.
Thank you, Elly. And I do like the engineering analogy very much. So it's a great pleasure to have Allison Bardin here with us. Professor Allison Bardin works on aspects of using Drosophila again as a model system, this Rolls-Royce of developmental systems. And she's made seminal studies in understanding how the gut stem cell niche, which we have ourselves, works. And I'm looking forward to hearing this illustrated.
So one of the fundamental aspects of stem cells is their dual capacity to both self-renew as well as produce terminally differentiated cells. And in healthy tissues, this process is kept in check and in balance. However, in disease context, this process becomes imbalanced. So for example, we know that during aging, tissue regeneration and tissue renewal is affected and becomes much more difficult to do than in young tissues. It's clear in the cases of hair, muscle, and brain in humans. We also know that, in contrast, this process also becomes deregulated in the context of cancer, where there's a surplus of self-renewing cells. And both of these processes have actually been linked to some extent to the acquisition of mutations in stem cells. So this is very clear for cancer, for example, and this has been proposed to be one of the causes of age-related decline. So this is something that we've become quite interested in studying in my lab, and that for which Drosophila is a very good model system. So the goal here is to really understand basic mechanisms of how stem cells mutate. And as I mentioned, we believe that this has important implications both for cancer initiation as well as for aging. So as Andrea alluded to, the fly is actually very homologous to many parts of the human, and you can find body parts that are identical or quite similar in both fly and human. We're focusing on the intestine, which has analogous parts to the human intestine. So the model that we use, as Peter mentioned, is the intestine of the adult fly. In the past 10 years, this has become really an excellent model to understand basic mechanisms of stem cell biology. There are around 1,000 multipotent stem cells present in this tissue. These stem cells have quite a simple lineage. They self-renew and differentiate to produce two types of terminally differentiated cells, both enteroendocrine cells and enterocytes. These stem cells respond very rapidly to changes in the environment, so it's an excellent model to understand how stem cells may be integrating cues from the external environment. And of course, there are important similarities not only with mammalian intestinal stem cells, but really going beyond that to other mammalian stem cell models. So we think that what we're learning here in the fly model will pertain to other adult stem cells. I think it's also important to note that the fly genome is around 15 times smaller than the mammalian genome. So this makes some types of genomic studies actually much simpler and cheaper in fly. So a number of years ago, we became quite fascinated by an observation that we had in lab, which was that we observed that in a proportion of our flies that we could detect spontaneously rising neoplasia. And we could show actually that this was due to the spontaneous inactivation of a tumor suppressor gene. And these neoplasia can be quite large and grow, comprising around 30% of the entire intestine. And we could use deep sequencing methodology to sequence this neoplastic tissue and compare this with two reference samples from the same organism to show that really in these neoplasia there is an inactivation of this tumor suppressor gene that's driving this neoplasia. So this, in recent years, in the last 10 years, it's become more and more apparent that this is really the normal state of adult stem cells, and that both in human somatic stem cells as well as in cancers, mutations are prevalent. So we think that this model system can really push us forward in terms of understanding this process. And importantly, there's really many questions here that are left to be addressed, and we do not really understand yet the extent to which somatic mutations may impact human health and may interface with different diseases. So the type of questions that we're interested in addressing in the lab are really what are the contributions of distinct types of mutation to how the somatic stem cell genome is changing, and how does that impact the aging genome? What are endogenous as well as exogenous factors that may contribute to stem cell mutation? And this is something that we can do quite easily and rapidly in the fly, as this is these stem cells are interfacing with the external environment. What are the impacts of stem cell mutation on aging as well as tissue health? And finally, I think, and a really important question is, can we do anything to actually prevent stem cell mutation, or perhaps change the selective pressures within a tissue to select against mutations which may be harmful? And for all of these, we think that the fly is really an excellent model system to address this in. It's quite fast. Aging occurs over a period of 6 to 8 weeks of adult life. It's relatively cheap to grow large numbers of flies and house large numbers required for aging studies. As Andrea and Peter already mentioned, it's genetically amenable, and this really will allow us to get mechanistic insight into the processes that are going on here and underlying somatic stem cell mutation. So I'll just go ahead and stop there, and we can move back to Peter. Thanks.
So thank you, Alison. Thank you to all our speakers. So I think we all perhaps learned a few things that were a bit surprising from that Drosophila have dormant stem cells that you can wake up, and luckily we're not going to see a 6-foot giant Drosophila larvae walking down the street. That was a good thing. That we can potentially use regenerative capacities of the axolotl and its amazing party trick of regrowing its limb as a sort of owner's manual of how to build or engender and engineer regeneration in our own capacity. And we learnt about the gut system in the fly. Who would have thought that the humble fruit fly would have a gut or intestine system with stem cells very similar to our own? But I guess I'm going to be a little bit controversial here and say we're going to hear a lot of things about stem cells at this meeting, and there are going to be a lot of people making bits of human tissue and that have amazing capacities, little brains, little guts, and the public might wonder, why will we need these animal systems now to study? Surely we can do this all in the dish and study stem cells in the dish. Now, of course, personally, I have responses to that I can talk to at the end, but I thought we might just start in order of speaking and sort of maybe enunciate why you think that the relevance of your system is still strong today and powering on. So we'll start with Andrea.
Right, well, I think that all of the different systems that people use to understand various aspects of science, they're all complementary. And I think one of the great advantages of working with model systems is we're often looking at the entire intact organism. So with respect to what we're interested in, being able to wake up stem cells in the brain, there are many signals that go from the organism to the brain to tell these stem cells to wake up. And so we're very interested in how different organs and tissues talk to one another and how the animal responds to environmental signals. And this is something that we can do uniquely in model organisms because we're working with the entire animal. Some of the things that people are studying in the dish are, it's a great system for looking at individual cells and cell-cell interactions, but I think when you want to start looking at the entire animal and how the entire animal's physiology works, it's very important to start looking at intact systems, and that's something that we can do in Drosophila, in axolotl, and in mice, in the various model organisms that we all work on.
And I think people would be intrigued to know that Drosophila had insulin and that it was a critical pathway for its activation of its stem cells and growth. So maybe you could talk about how that pathway is conserved and a little bit in its action.
Yeah, absolutely. Well, I think when we think about insulin, most people will think about the pancreas and what we would call systemic insulin that can work throughout the body. And indeed, Drosophila has systemic insulin, which regulates the growth of the organism. But what we discovered, which was very interesting, is that there's another type of insulin which is actually present within the brain, and it's produced by another type of cell called glial cells, which actually envelop the brain, and they secrete this other type of insulin which is what's responsible for waking these stem cells. And these insulins have homologs in vertebrate systems, and indeed it's been shown that insulin-like growth factors can stimulate stem cell proliferation in mammals. And so again, we think there are these parallels, these really interesting basic mechanisms that are conserved from lower organisms to higher organisms.
So perhaps a paradigm shift of insulin signaling biology that might not have been made by studying human stem cell interactions.
Yes, absolutely.
Yeah. Fantastic. So Elly, we've seen the aeroplane, and I guess of all the systems, that's the one where you think, well, really, we can't do that, and therefore we have to look at that. Do you think the engineering principles will get there, or will this mystery be cracked, or will it forever elude us?
Yeah, when I started the studies, I was a basic biologist and was really just fascinated by the process of regeneration. As we have learned more and more about the principles of how a salamander can regenerate its limbs, surprisingly, the most skeptical person, me, does feel that maybe one day it could be possible. Of course, it's very complex. There are many changes that have occurred in the genomes and of course then the cells and tissues of mammals as over evolution. But I do feel that through the study of the salamander, the basic principles of how a limb should be built are coming forth, and that the salamander will continue to give us information, for example, of how to build a limb at an adult scale. For example, because these animals build the limb in a very tiny embryo at first, and the same process works in an adult salamander that has an adult amputated limb that's 10 or 20 times the size of this small embryo. So I think that it will be complex to make sure that the immune system, the blood system, the nervous systems, all these cells are interacting properly to induce a limb to regenerate. But as I said, I think the combination of engineering cells in vitro plus interfacing with the natural biology of ourselves may allow complex regeneration to occur. Of course, we can never promise whether a limb will be regenerated, but I'm surprisingly more hopeful than I was when I started.
And recently, Elly, you've had some technically enabling features in your system come to fruition after many decades of hard work. Would you like to just illustrate how technical advances can advance your understanding?
Yes, so in our system, it was very important to develop methodologies to be able to manipulate the genome and DNA in order that we could, in a very refined way, control the activity of proteins in regenerating tissues. And so therefore we developed approaches to be able to put a piece of DNA and produce proteins in certain places in the animal, either by injection into the embryo or by the development of viruses that can infect certain tissues in the regenerating limb. And this has given us the precision with which we can really assign a function to a certain gene or protein in the process of regeneration. And actually, such developments in our system are analogous to the types of gene therapy approaches that people are developing in humans and that are really relevant for advances that we're seeing now in stem cell research.
Yeah, thanks, Elly. So, Allison, in terms of your stem cell system, it seems directly analogous to the human situation. How much do you think you can push that analogy, and where do you think you find the barriers in understanding to translate, and how much of your interest is in actually finding general principles, as you alluded to, the fact that you can use it to define how adult stem cells function generally, and how much do you think will be directly transferable to the neoplasias that we find in our gut system?
Yeah, I mean, I think I think a lot of things will be directly translatable, and the types of mutations that we're finding are really very, very similar to what occurs both in the context of human cancers, but also in the context of developmental abnormalities in humans. In a lot of instances, we really don't know molecularly how what's going on here. So what are the erroneous repair mechanisms that would be repairing in this context? And what we can do in the fly is to really go in with genetics and inactivate a gene and ask, how does that impact the mutational spectrum? I just wanted to also go back for a second to what you were mentioning about the organoids and raise a couple of points that I think are important there. So one thing I think that's been very exciting that's come out in the last couple years in my field, looking at the intestine, is really a better understanding of how organ-organ communication impacts stem cells. And this is also a point that Andrea brought up. And I think that this type of niche signal would be really lacking in an organoid-type context. So I think that's a context in which, in the fly, we can really push forward to understand that. And we also have genetic tools to really manipulate different tissues very precisely in the fly. So I think for understanding really at an organismal level, as Andrea mentioned, how signals can control stem cells, this will be very important. I think another important point has to do with the genetics, which Elly brought up as, understanding genetics and using genetics is extremely powerful in these model systems. But in the mammalian organoids, we're somewhat confounded by all of the analogous genes. So if you would like to inactivate a given pathway to understand how a pathway works, many times there's a lot of redundancy built into the mammalian networks. So this is actually something quite difficult to address in the mammalian context, where more simplified genomes are better in a sense to really identify all of the components in a given pathway. So I'm hoping that I think there obviously will be important analogies that we can draw. I think at some point that we will not be able to push any further with that. So with respect to the types of mutations that we're seeing, I think they would be similar to quite simple types of cancers. I'm not sure that they're going to mimic very complex, highly mutagenic late-stage cancers, or very metastatic, very late-stage cancers. So the analogy will probably stop there. But in terms of understanding the initiation process and processes that can really drive the initiation, I think it's a great model system.
Yes, yes. And the other thing you touched on is aging, and I think a lot of the public would be surprised that flies age. Certainly the ones that they're used to seeing.
We all age.
And they seem to live fast and die young, but that's not at all true.
About all of the organisms that you mentioned that you're working on, in terms of the history of looking at them and researching the research that's been done in the past, and then where we are as far as what we've been learning, and have we learned a lot, if you could each talk about the organism that you work with. Also, how the funding lines up for research on the organisms that you're talking about, or in general on model organisms. Is it difficult? Is it a challenge to get research funded? And how has that been historically as well?
I think it's always a challenge to get research funded at the moment. I think it's also fairly country dependent from what I understand. I think luckily in France, thus far, they've been fairly supportive of model systems, including the fly. I've heard from colleagues in other countries that they are feeling pressure to incorporate mammalian models into their grants. I think in terms of serving the public, it's important to have models that are relatively cheap, where basic principles can be described without having to get into very costly mouse experiments that really explode the budget. So I think just from a basic biology point of view, I think it is important to still fund these basic model systems.
Yeah. I would agree that it is very country specific. I'm from the United Kingdom, and we have funding from the Wellcome Trust, which has always been very open to what we would call discovery science. And I think if you look back through the years, and look at basic, well, discovery science or basic research, there are so many advances that have impacted on human health over time. And so I tend to think of it as a continuum. I don't really separate translational research from basic or discovery science. It's a continuum. And as I mentioned before, I think all different systems that we work on, they're complementary. They all fill a particular gap in our knowledge. So I think it's very important to look at the breadth of science that's being done and ensure that there's support for all different aspects because they influence each other. Coming back to the start of your question about, Drosophila has been studied for well over 100 years now. And I think that when what people often forget, especially if you're reading papers on mammalian systems, a lot of the genes they're studying were discovered in Drosophila. Things like wingless, which is Wnt, engrailed, the Hox genes, these all came out of Drosophila research. Sometimes the names are the same, sometimes they've been changed. But it's actually remarkable, even in my lifetime, to see how much work on Drosophila has influenced work on higher organisms and human beings. And they're often things that you couldn't have predicted. So I would like to see that funding is maintained across the board because we cannot always predict where the discoveries are going to come from.
Maybe I can just reinforce that. So one of the proteins that I spoke about, sonic hedgehog, is exactly one of these proteins that was identified in Drosophila with people just trying to look at cuticle patterns, just the pattern of the skin in Drosophila embryos. And then it turns out that the sonic hedgehog is very important for the limb development, and this was elucidated by studying chicken and mouse embryos. It's also extremely important in medulloblastoma, which was really worked out in mouse embryos, and now this protein is extremely important in limb regeneration in the axolotl. And so there you can see, exactly as Andrea pointed out, that we need a community of researchers working at every level in different organisms that have different strengths for identifying important factors, and that these factors are used over and over again in different biological contexts for developing the brain, for developing the limb. So we are really cutting off our nose if the funding bodies don't continue to fund research in basic model organisms.
Yes, and I think looking at that axis of development, the hedgehog signaling pathway is very illuminating because as it was said, it was discovered in Drosophila, but when I did my postdoc, it was in a lab that was a Drosophila lab that then cloned a lot of the vertebrate hedgehog genes and then looked at their activities. And a lot of people are extremely shocked that they had very similar inductive activities in vertebrates. That was, that's within my scientific lifetime, let alone my actual lifetime. So you can imagine the impact of finding a similar finding now. So that pathway's gone on to be one of the central pathways in development, but also the central pathway in cancer biology, and the activities that were described then and the drugs that were used to modulate that pathway are now treating cancers in the clinic. So this is the important, I think, I prefer not to make a distinction between model and non-model systems. It's where we have a problem, and this is we're not necessarily immune from it in Australia, in fact, is things that are immediately translatable and obvious to ones that are a bit more blue sky. And I think people often struggle to invest in those things that have no obtuse or obvious impact on human health. But yet those are the things, if you look back through history, that have changed the human condition most dramatically. It's not the obvious step of doing the next obvious thing. Of course they need to be done because you're translating findings you've made already. But where is the next big discovery going to come from? And then understanding how nature has generated tissues and organ systems has been a recurring well for that type of inspirational science. So if we don't keep investing in it, we won't have more of the types of similar activities and drugs that have been developed from these developmental systems in the past. So when I moved back to Australia, it was clearly a struggle to convince people that zebrafish were in a fairly mammalian-dominated system where the physiology is much more directly relevant to humans, that zebrafish could tell anybody anything about the human condition. But I think all we can do as model organisms is make these types of statements and illustrate with the quality of our science the fundamental discoveries that we're making. And then I find colleagues over time begin to accept your system for what it is, a powerhouse of discovery for which they can then utilise as a toolbox for their own research.
Just to highlight that, I mean, one of the prime examples of that today is the CRISPR-Cas9, which we'll hear about on the last day of the talk.
Yes, and I think we've all experienced revolutions in our own systems over a lifetime. That we're talking about them like they're static things, but we've heard about Elly, about the genetic development, the genome, that she's just so proudly, been able to finally join the genome club, which is fantastic. A very difficult genome in the axolotl. For us in zebrafish, it's been the power of imaging, and also in Drosophila, these amazing new imaging systems, the lattice light sheet, the normal light sheet, super resolution microscopy, AIRI scanning. All of these things increase our ability to peer into the living tissue in the organism and make seminal discoveries. And our systems have been taking advantage of all these techniques. CRISPR-Cas9 has completely changed the way I do my science. So these things, we're not talking about organisms that we do the same thing and we just get the same results. We're continually evolving our systems as well. Right, I think that's pretty much dead on time. Oh, has the audience got any more questions? Well, I'd like to thank all the panellists for turning up at 7:30 in the morning on a cold Melbourne morning. I apologise for the weather in Melbourne, but this is the midwinter and this is as bad as it gets. And I hope you enjoy the rest of the meeting, and thank you for sharing your insights with us.
Thank you.